The overall goal of this software-based protocol is to accurately and rapidly estimate total body biological radiation dose from dicentric chromosomes found in sets of automatically filtered metaphase cell images using machine learning-based image processing techniques.
This method can help answer key questions regarding the response to a mass radiation exposure event. Timely examination of a large number of samples will be necessary to determine radiation dose.
The main advantage of this software is automation for image filtering, calibration curve generation, dicentric chromosome detection, and the radiation dose estimation. These are traditionally time-consuming and labor-intensive tasks.
Though this method can provide insights into radiation cytogenetic biodosimetry, it could be applied to select the best chromosomal images for diagnostic analyses of inherited and acquired genetic diseases.
We first had the idea for this method when we began developing chromosome image analysis methods for automatically scoring fluorescence in situ hybridization images. Colleagues had indicated that the current capacity for testing multiple exposed individuals was also very limited.
Open the software and create a new sample from the menu. Then, select the folder containing the metaphase images to analyze. At the prompt, supply a unique ID to identify the sample. Only alphanumeric, underscores, and dashes may be used. Then, provide a description of the sample if desired.